Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, in yesterday's class, we learned about genomic imprinting, in which we discuss about certain imprint control regions such as the KCNQ1 locus as well as the H19 IGF2 cluster. Today, we are going to see if certain things goes wrong in those clusters, how they result into a syndrome known as Beckwith-Wiedemann syndrome. So we are going to learn about Beckwith-Wiedemann syndrome in detail. This is also abbreviated as BWS. Okay. This syndrome is characterized by overgrowth, which results into macrosomia, means the large body size at birth, as well as so it leads to the bulky and gigantic body. So the fetal and postnatal overgrowth is seen in this disease, as well as it leads to the macroglossia, means the larger tongue size, hemihyperplasia, the asymmetrical growth of one side of the body, and this also leads to predisposition to childhood tumors such as the kidney cancer and lung cancer. So this Beckwith-Wiedemann syndrome, this happens when the imprinted regions such as the KCNQ1 locus and the H19 IGF2, either of them are abnormal or both of them are abnormal. If certain thing goes wrong in those, then they result into this Beckwith-Wiedemann syndrome. So first, before discussing about what goes wrong, let's discuss about those clusters. So what we learned yesterday was KCNQ1 locus and the H19 IGF2 cluster. Okay, so let's discuss about KCNQ1 locus first. The maternally inherited genome means the maternal allele. Okay, in the maternal allele, the KCNQ1 gene is expressed as well as the CDKN1C gene, which produces the inhibitor of CDK cyclin dependent kinase that is also expressed in the maternal allele. But the paternal allele, it expresses KCNQ1 OT1 long non-coding RNA, which is responsible for the suppression of CDKN1C. So it leads to the silencing of CDKN1C. That is why this inhibitor of cyclin dependent kinase, it is not produced by the paternal allele. Okay, so we can say that maternal allele, it leads to growth suppression. Okay, and the paternal allele leads to growth promotion. Okay. In the H19 IGF2 cluster, this contains two genes, the H19 gene as well as the IGF2 gene. The H19 gene it codes for the non-coding RNA H19, while the IGF2 is the growth promoting factor. So the maternal allele, in case of this cluster, it does not produce IGF2 because it has this cluster has the CTCF binding site which is unmethylated in maternal allele, and that is why CTCF can bind to it, and the enhancer which control the H19 as well as IGF2, it does not have control over IGF2, and that is why IGF2 is not produced. So only H19 gene is expressed, okay, which produces the H19 non-coding RNA. This is a non-coding RNA, which also leads to growth suppression. While unlike maternal allele, the paternal allele has the methylated CTCF binding site as well as the H19 gene is also methylated. So there is no H19 expression. Instead, there is IGF2 expression, which is a growth factor, right? So this leads to growth promotion. Okay, so I hope from this it's clear that which allele does which job. Okay, So, when we learned about the characteristics of Beckwith-Wiedemann syndrome, I told you that it leads to overgrowth. So now, from this summary, it is quite clear to you all that what could lead to this syndrome. Pause the video and think about it. So, if the Beckwith-Wiedemann syndrome, it is going to lead, it is going to result into overgrowth. This means that the growth suppression genes should be silent and the promoting genes or the growth activating genes, they should be overexpressed. The expression of those genes should be elevated. So, let's list down the causes which lead to the Beckwith-Wiedemann syndrome. Okay, so let's list down the causes of Beckwith-Wiedemann syndrome one by one. So this is a genetic as well as epigenetic disorder, means certain mutations in certain genes can lead to the Beckwith-Wiedemann syndrome. So first let's list down the genetic causes. So mutation. So where in which gene in which sorry the which mutation could lead to BWS? Mutation of growth suppressing gene, because if there is no growth suppressor gene factor, the grow it will automatically lead to the imbalance and there will be overgrowth. So that is why mutation in CDKN1C gene, which is inhibitor of cyclin dependent kinase. Okay, so this is a growth suppressor factor. That is why if there is mutation in this gene, the gene producing the CDKN1C, that will cause the Beckwith-Wiedemann syndrome. So this is a genetic cause. Now let's list down the epigenetic causes. The epigenetic causes could be that if certain DMR is not methylated. So if the DNA methylation, if there are certain abnormalities in DNA methylation, it could lead to BWS. Let's see how which abnormality in DNA methylation can lead to BWS. So in the KCNQ1 locus, the DMR which is methylated, that is the KCNQ1OT1. So this is an intronic region, which intronic region of KCNQ1 gene, okay, and this codes for the long non-coding RNA, and this is methylated in case of maternal allele, and that is why we say that KCNQ1, this is maternally imprinted gene. Okay, this is maternally imprinted region, while the H19 IGF2, this is paternally imprinted region, means the CTCF binding site as well as the H19 genes, those are methylated. Okay, so if there is expression, there is more expression, elevated expression of KCNQ Q1OT1, or if there is suppression of CDKN1C, that could lead to Beckwith-Wiedemann syndrome, or if there is suppression or silencing of H19 gene in both of the alleles, or if there is overexpression of IGF2, both of those cases, all of those cases can lead to the Beckwith-Wiedemann syndrome. So we can say that if there is no DNA methylation, it is absent in maternal allele at KCNQ1OT1 at this DMR. So what happens that if this is not methylated, means this will not be this will not be suppressed. This means it will lead to KCNQ1OT1 production from maternal allele as well as the paternal allele. So if both of the alleles, they are going to express the long non-coding RNA, which is going to do the suppression of CDKN1C, this will eventually lead to the no production of CDKN1C, which is a inhibitor for cyclin dependent kinase, and that is why there will be more cyclin dependent kinase which is going to result into the overgrowth. Okay, or if DNA methylation is absent at the CTCF binding site Okay, no, no, no. If the DNA methylation, it is present at CTCF binding site of, let's say, the H19 IGF2 cluster. So at this cluster, the CTCF binding site, if it is methylated, it means that CTCF won't be able to bind that. And this will lead to the controlling of enhancer. Enhancer can control both IGF2 as well as H19. So this will lead to the expression of IGF2, and let's say that there is methylation at H19. So let's say that DNA methylation is present at CTCF binding site as well as the H19 gene. In both of those cases, there will be more production of IGF2. The IGF2 production will be elevated because it is now going to be produced from both maternal allele as well as paternal allele, but there will be no expression of H19, which is a growth suppressing non-coding RNA, okay, which has the role of suppressing growth, and that is why this can lead to the Beckwith-Wiedemann syndrome. So the DNA methylation abnormalities that I listed down here, that could lead to the Beckwith-Wiedemann syndrome. But again, there is also a thing which there's also a phenomena that we call as uniparental disomy, which can lead to Beckwith-Wiedemann syndrome. Okay, so uniparental disomy means, imagine that this cell which has a maternal copy of genome as well as a paternal copy of genome, this is undergoing the mitosis. Okay, so this is undergoing the mitosis, and during division certain thing goes wrong, and instead, so the daughter cells should have uh, the composition of this composition, something like this. Both of them should have a maternal copy of genome and a paternal copy of genome. Okay, so both of them should have a maternal chromosomes and paternally inherited chromosomes. But if certain thing goes wrong, and instead of this, the mitosis results into, so the chromosome distribution in daughter cells, that goes wrong, and it results into this, let's say this. Okay, means there are three chromosomes in one daughter cell, and there is only one chromosome in other daughter cell. Now this is the result of mitosis, this imbalance. Okay, so there will be degradation of one chromosome in this cell. Okay, so this cell will degrade a chromosome for balancing for maintaining the balance. So if there could be two possibilities. First, that it could lead to, it could degrade the maternal copy chromosome, or it could degrade the paternal chromosome. So there are two chances, two possibilities. First, that if the one paternal copy is degraded, it will lead to a normal cell. So this is a normal cell, okay, which has a maternal chromosome and a paternal chromosome. But by chance, if there is degradation of maternal chromosome, then it could lead to a cell which has inherited both copies, which are inherited from father. So it will have only paternal chromosomes. Okay, so such condition is known as uniparental disomy. In this case, it is unipaternal disomy. This is unipaternal disomy, or paternal uniparental disomy, call it whatever. So this unipaternal disomy means that the cell has inherited both of the copies from the father. And there could be another case known as unimaternal disomy, means both of the copies of the genome, they are inherited from mother. So this abnormality can also cause Beckwith-Wiedemann syndrome. Let's see how. So let's say that there is unipaternal disomy, means both of the alleles, they are paternal alleles. Okay, so what happens in the paternal allele? Let's combine both of the cluster. Okay, In the paternal allele, there is expression of KCNQ1OT1 long non-coding RNA, okay, which is going to suppress the expression of CDKN1C, okay. And in the same chromosome, which is inherited from father, the paternal allele, it has the IGF2 gene, the CTCF binding site. This is methylated, as well as the H19 gene. This is also methylated. Okay, and both of the genes, they are controlled by this enhancer. So now those enhancers, so this enhancer, it can control IGF2 because there is absence of CTCF, and that is why there will be production of IGF2, but there will be no production of H19. Okay, so in the unipaternal disomy, so this is the paternal allele. Okay, the exact opposite things happens into the paternal allele, but due to the unipaternal disomy, both of the alleles will be paternal alleles. So let's say that this thing is repeated. So both of the genes, they are going to express the KCNQ1OT1, which is going to inhibit CDKN1C. Both of them are going to express IGF2 and suppress H19. So what will happen in unipaternal disomy in this case will be that there will be no CDKN1C. Okay, CDKN1C. Okay, there will be no CDKN1C. There will be no H19. Okay, and there will be elevated expression of IGF2. So IGF2 expression will increase. So IGF2, which is a growth factor, this will lead to overgrowth, and as there is no H19, which is a growth suppressing non-coding RNA, as well as CDKN1C, both of them suppress growth. So absence of this factor will lead to the imbalance, and that is why this is how the unipaternal disomy will result into the Beckwith-Wiedemann syndrome. So I hope with this, what causes Beckwith-Wiedemann syndrome and what are the characteristics of that syndrome, that is clear to you all. So there could be some genetic reasons for this disorder as well, but we wanted to focus on the imprinted locus. What you think, what goes wrong? So to list down the epigenetic causes of this disease, first is that if the DNA methylation, there is DNA methylation abnormalities, such as either the DNA methylation can be absent on the maternal allele at the KCNQ1OT1, or the DNA methylation can be present on this H19 gene or the CTCF binding site. The other epigenetic cause for this syndrome is unipaternal disomy, which will result into the inheritance of two paternal alleles instead of a maternal allele and a paternal allele. Okay, so let's say that the epigenetic causes are, they are: first, if there are certain DNA methylation abnormalities, okay, or if there is unipaternal disomy. The genetic causes could be if there is mutation in CDK N1C gene, means this is not expressed. If there is mutation in H19 gene, let's say that this is also suppressed. If there is mutation in IGF2 gene, such as the expression is elevated, okay, this is rare case, but the CDK N1C gene mutation and H19 gene mutation, they can lead to BWS. So with this, we are done with today's lecture. In next lecture, we will learn about one more imprinted cluster known as SNRNPN cluster. Okay, we will learn about this epigenetic control region, as well as we will learn about the Prader-Willi syndrome and the Angelman syndrome. Till then, see you. Bye bye.